thanks everybody for coming. It's John Snow's birthday today, so isn't it great that we've got George Davey Smith, one of the best epidemiologists in the world, that started off in Scum Auxiliary, as has been pointed out to you, but that hasn't been so successful. You can get some of the music on the anthology Obscure Independent Classics, the special Hamster edition in 1988. So if you're interested in that, that would be good. So George is an epidemiologist and is always at the front of the field. He published a book about systematic reviews really before Cochrane got going. He's come here today and he's going to be talking about some of the latest advances around Mendelian randomization, epigenetics, which is what he has been advancing in recent years. So thanks so much, George, for joining us.
Thanks very much, and it's a pleasure to be here. So I'm going to talk just about a few topics around how genetics and genomics might actually have relevance for population health. I'm going to talk about how genetics can help strengthen causal inference about factors that genuinely have an effect on population health. I'm going to talk about using epigenetics for exposure assessment and prediction. And finally, I'm going to talk about how considerations of genetics helps us understand the prevention paradox, Jeffrey Rose's prevention paradox. So I'll go relatively superficially through 4 topics, but I'll spend most of the time on the causal inference. Now, if you're interested in environmentally modifiable exposures, behavioral factors, diet, or external environmental exposures, the obvious thing to do would be to measure the exposures themselves and see how they influence people's health, how they relate to people's health. But the problem with that approach, I think, is brought home by this advert in the Boston Globe. The average American lifespan has increased nearly 3 years over the last 2 decades. We've been selling vitamins at a discount since 1977. Coincidence? We don't think so. So here, vitaminshop.com, who might have got a pecuniary interest, I guess, in this issue, want you to believe that it's because you've been taking their vitamin supplements that life expectancy has improved. Now, the reason they could say that is because there was what at the time appeared to be very reliable evidence from observational epidemiological studies that vitamin supplementation was reducing the risks of some chronic diseases. And the sort of evidence that fed into that was, for example, this classic paper with many thousands of citations from the health professionals follow-up study, where they did an epidemiological study asking people if they took vitamin E supplements and how long they'd been taking them for. So the important thing here is that what the exposure that was being examined was having the supplement, putting it in your mouth and swallowing it. That is the exposure being examined. So that is obviously something that can be tested in a randomized trial, unlike most exposures, or many exposures can't be that directly tested in observational epidemiology. But what they showed is, they measured all the other risk factors and they adjusted for them in the statistical ways that you do. And they found that there was a so-called independent effect, that vitamin E supplement use was associated with around a 30 to 40% lower risk of coronary heart disease. They validated this in a separate study, the nurses study, which showed the same findings. So they got this in two large-scale studies. The New York Times reported it as vitamin E greatly reduces the risk of heart disease, studies suggest. So that's makes the translation directly from the epidemiological observation to this actually having an effect, i.e. reducing the risk of heart disease. And you'll be unsurprised to know that when the large-scale randomized controlled trials testing exactly the same exposure but in a trial situation were carried out with up to 13 years of follow-up, they showed absolutely no effect of vitamin E supplementation on coronary heart disease. Now, from a public health perspective, around 50% of folk in the US at the turn of the century were taking vitamin supplements which contained vitamin E. either multivitamins containing vitamin E or vitamin E on its own, which took off after those studies appeared. And so this, at least it's not killing people. It was sort of neutral in its effect. But but people taking things which aren't actually benefiting them can of course have downsides. For example, they're not going to necessarily take all the other measures which will reduce risk because they think they're reducing their risk 30%, 40% by taking the vitamin E supplements. Now, the reason these observational studies got things wrong are very likely that the taking vitamin E supplements is just so strongly confounded with other factors, socioeconomic, behavioural, physiological factors, and you just simply cannot take those into account through statistical adjustment or statistical modelling. Whatever people tell you about marginal structural models and G estimation, etc., somehow magically recovering causal effects from observational data, they don't work in most circumstances. And the real problem is when they might work, you don't know how to separate the times that they might be working from times that they don't work until you've got data from things like trials. So conventional observational studies, particularly of confounded factors related to many other exposures like dietary factors, but like occupational exposures, external environmental exposures, etc., are very difficult to study in the observational context. And also having biological evidence from cellular models or animal models, etc., has been shown to be extremely fragile in terms of how much it strengthens evidence. So, so much for the antioxidants, but, other people have said, well, so much for observational epidemiology. This is a US cartoon where the newsreader presses the button and says, according to a report released today's random medical news, the wheel spins and coffee causes depression in twins. That's today's news story. And of course, every one of these combinations is, of course, spurious, except red wine does cause sexual dysfunction in men aged 25 to 40, although it's an extremely distant memory for me now. That so given the problems with straightforward observational evidence, one thing which the genomics revolution really has given to epidemiology is the ability to do essentially randomized analyses within observational data. Because the way that genetic variants are sorted in the population according to Mendel's first and second laws leads to them not being associated with confounding factors in the way that actually taking vitamin E supplements is, or taking vitamin C supplements, strongly associated with confounding factors. There's a very good genetic variant which mimics vitamin C supplement use, which is in the human vitamin C transporter in the gut. And people who carry copies of this variant have lower vitamin C by about a third of a standard deviation. That variant, as we and others have shown in huge populations, is just not related to any confounding factors. Whereas taking vitamin C supplements, of course, is hugely related to confounding factors. And so this essentially random distribution of genetic variation, as long as you take population stratification into account, and you have to work very hard not to take it into account. You have to, deliberately ignore it and not use the methods that are available for taking it into account. As long as you take that into account, i.e. different ancestral origins, the genetic variants are not confounded, and nor, of course, are they influenced by the disease processes. A major problem in epidemiology is reverse causation. It's that the early stages of disease influence your exposure or parent exposure. So atherosclerosis, which is an inflammatory process, increases every acute phase reactant, CRP, fibrinogen, interleukin-6, and it's no wonder that all of these things strongly predict coronary heart disease. So you get reverse causation. So Mendelian randomization can be considered as, in some ways, analogous to a randomized controlled trial. And here's a situation where we make that analogy explicit, that observational epidemiology combined with biological evidence suggested that selenium could reduce the risk of prostate cancer. And the evidence was seen as so good for that just like it was for vitamin E and coronary heart disease, that huge amounts of money was spent on randomized controlled trials. One huge randomized controlled trial called the SELECT trial, randomizing men to a selenium supplement which increased their plasma selenium. That trial carried on for many years at the cost of tens of millions of dollars, and at the end of the trial, what they showed was that increasing selenium through supplementation had no effect on prostate cancer incidence. Now, in the Mendelian randomization paradigm, you have genetic variants which relate to selenium level. You can use either single variants in this approach, or if there are multiple variants, you can use the multiple variants to increase your statistical power and to allow you to test some of the assumptions. So these variants exist that relate to selenium levels, and then if you take those genetic variants that relate to selenium levels and you look at them in the large-scale case control studies of prostate cancer, you get an estimate of how much selenium might influence prostate cancer risk. And we did that with 22,000 cases and 22,000 controls from a large-scale consortium called a prostate cancer called Practical, and our findings were the same as suspiciously similar. Those honestly are the findings to those of the randomized controlled trials. And importantly, the confidence intervals are pretty much the same as well, because you can get these huge samples. And my guess is that if these data had been available, people would not have launched the trial, and we'll come on to that later in terms of how Mendelian randomization has stopped trials being launched. A couple more examples. Body mass index protects, observationally, appears to protect against lung cancer. This isn't just the very thin people having increased risk of lung cancer, it's right the way across the board, such that according to WHO categories, the overweight have a higher risk than the obese of lung cancer. It's just linearly across the board. And the evidence was considered so strong that the World Cancer Research Fund listed being thin as a possible cause of lung cancer. But the Mendelian randomization evidence suggests that this is not a causal association, that it's more likely to be due to the fact that smoking reduces your body fatness, and also very early stages of disease can lower body mass index. And I mentioned C-reactive protein. A huge amount of medical research has been done on this protein. It's an acute phase reactant related to the inflammatory process, and it's been implicated in virtually every disease you can imagine, including lung cancer. And observationally, C-reactive protein predicts lung cancer risk, and that's after you adjust for smoking and socioeconomic position and lung function and whatever else you can adjust for. And yet the Mendelian randomization evidence suggests that this is entirely non-causal. So putting this into a different language than randomized controlled trials, econometricians introduced the method of instrumental variables, whereby you have a variable which acts as though it were randomized, i.e. it's something which is not associated with confounders. It has no effect on the outcome except through your exposure of interest, but it does relate to your exposure. And genetic variants can be excellent instrumental variables. So if you have a genetic variant, in this case it is a cis variant in the CRP, in the actual gene that produces this protein near the promoter region, and people who carry one version of this variant or have higher CRP levels. And then if you group people according to their genetic variant and you carry out an analysis, it's like an intention to treat analysis in a randomized trial. Of course, there's huge overlap in the CRP between the groups, just like there is in a blood pressure in a randomized trial of the people on the placebo and on the active treatment. But you get groups with reliable difference in your exposure. So your instrumental variable is your genetic variant, which relates to your exposure, in this case C-reactive protein. And by relating your instrumental variable both to the modifiable exposure and to the outcome, in this case coronary heart disease, that allows you to estimate both whether this is likely to be causal, and secondly to get some estimate of the causal effect by simple ratio measure in the simplest case. and that allows you then to make this estimate of this causal effect. And in the case of C-reactive protein and coronary heart disease, that estimate is null. There was so much interest in CRP that many pharmaceutical concerns were trying to develop drugs to lower CRP, but the Mendelian randomization evidence stopped that development from carrying on. So just to reiterate about instrumental variables, the genetic variant must be associated with the exposure. The genetic variant is independent of your confounders, and the genetic variant is independent of the outcome if you properly took into account the exposure, i.e. all the effect of the genetic variant on the outcome is mediated through the exposure. Those are the assumptions that you are making, and we'll come back to those. Now, the genetic instruments can proxy for a wide variety of modifiable exposures, environmental factors including dietary factors, early life factors, birth weight, child obesity, puberty onset, lots of endogenous biomarkers, now very, very large numbers of metabolites, some proteins, some proteomics and metabolomics, very particular drug targets, I'll come back to that, and then epigenetic markers such as DNA methylation and other genomic measures like transcription factors. So you can test all of those in MR studies, and the number of actual Mendelian randomization studies has sort of taken off, partly, in fact largely, because the genetic instruments are being identified by the ongoing march of genome-wide association studies and the larger and larger samples that are available for such genome-wide association studies. One area of particular interest is when you can use Mendelian randomization for predicting the effects of manipulating particular drug targets. So I already mentioned CRP, where the Mendelian randomization had turned interest away from developing this as a therapeutic target, but to situations like PCSK9 monoclonal antibodies, which lower cholesterol, which have recently been licensed on the basis of genetic data, essentially on short-term trials, without the long-term trial, event trial data, have been licensed. And you can predict generalizability of treatment responses because you can't do trials in every single group. But if you have genetic variants which proxy for the pharmaceutical intervention, those can be looked at in many groups because genotyping is cheap compared to actually randomizing people and following them up for years in a randomized controlled trial. So I think it'd be fair to say that this approach will be used actually to prioritize which things actually then do go forward into event trials where it's worth putting the expense of an event trial. There's many cases of where event trials have gone on at the cost. Some of these really are the cost of $100 million trials, at the cost of huge amounts of money, in situations where the genetic data, if properly looked at even at the time, in some cases the trials were set up, were discouraging. So, for example, there are a variety of activated phospholipases. There's secretory phospholipase and there's LP-PLA2. They're related but to different molecules. There's drugs which target these. There's been several very large-scale trials which have produced no effect on the outcome. But the Mendelian randomization studies also just predict no effect. And there's one, gene study could have preempted VISTA-16. One area where there's been many trials is raising HDL cholesterol. You'll remember HDL cholesterol is good cholesterol. We were always told that raising HDL cholesterol was a good thing. The Mendelian randomization or genetic evidence suggested that wasn't the case, that HDL cholesterol is not protective. Just circulating levels of HDL cholesterol on their own are not protective against coronary heart disease, whereas LDL cholesterol definitely increases the risk of coronary heart disease. But a lot of trials went ahead with an absolutely null. They made big effects on HDL cholesterol, but no effect on events. And here they have effects like on GSK's heart drug misses primary goal. And here's an instrumental variable study telling us that insider trading happens. this is the day, this is when officially that GSK trial was released at a conference, but a day before the share price plummeted. So some people in the know were flogging their shares before the trial happened, unless this is some remarkable coincidence. Now, I'm just going to run through a few slides. I'm going to run through these quickly, given the time, but this is recently published in the New England Journal of Medicine by Brian Ferentz and colleagues. This is using genetic variants for very particular mechanisms of drugs. So first, this is looking at HMG-CoA reductase, which is, of course, the mechanism of statins. And you have a genetic variant which mimics inhibition of HMG-CoA reductase in the HMG-CoA reductase gene. You can combine several variants to create a score and put those into quartiles. And what this shows is that if the people carrying the variants associated with less and less HMG-CoA reductase activity had lower and lower risk of coronary heart disease. And then this is the predicted effect on coronary heart disease of a particular difference in LDL cholesterol. Another drug which has been introduced much more recently that lowers cholesterol, and which was developed because of genetic data that knockouts for this gene essentially had a bit large effect on coronary heart disease. And this is PCSK9. This is inhibitors, but genetic variation related to less PCSK9 activity have the same effect, the same dose-response effect on lowered rates of coronary heart disease. And the same predicted effect on coronary heart disease for a given reduction in cholesterol. So however you lower your cholesterol, this suggests you're going to get the same effects. These effects are about twice the size of the effects you see in RCTs of statins for a given difference in cholesterol, which is precisely what you'd expect because the genetic variants have been associated with lifetime differences in cholesterol. Whereas in a statin trial, you're taking it for, say, 5 to 7 years. People have been randomized. But, atherosclerosis starts early on. And so the genetic variants tell you what in public health terms could be achieved by any policy which led to very long-term lowering of cholesterol levels in people in populations. You can then use the Mendelian randomization to predict the combination of the drugs because the event trials going on for PCSK9 inhibitors, because they're so expensive, it's people who are either are said not to be able to take statins, although it's exaggerated now, who can't take statins, or whose cholesterol isn't controlled even with a statin, so you add it on top of a statin. So the trials are adding PCSK9 to a statin, but the Mendelian randomization evidence, this is before the trials have appeared, suggests that the PCSK9 inhibition will have an effect over and above the statins to the extent to which it leads to further cholesterol reduction. In the randomized controlled trials of statins, there was a small increase in the risk of type 2 diabetes. Now, for virtually every risk group, this was heavily outweighed by the beneficial effect in terms of reduction of coronary heart disease. But there was a small increase of type 2 diabetes, and the Mendelian randomization supports this as actually being causal, i.e. on target. This doesn't appear to be a sort of off-target effect of the drugs, it appears to be through the mechanism. But very importantly, because it was suggested that PCSK9 inhibition in some ways might be like clean cholesterol lowering without this effect, you see exactly the predicted effect from the Mendelian randomization studies is precisely the same. If you're lowering this through an LDL receptor mechanism, you get the same effect on type 2 diabetes, and it's combined if you influence both mechanisms. So what you see here is you can use Mendelian randomization to predict the on-target effects you want, but you can also use it to predict on-target effects that you don't want, i.e. adverse effects. And because a genome-wide association study data exist on so many endpoints, you can actually look to see how manipulating any particular target is likely to influence 100 diseases and 1,000 traits. You can actually look right the way across the board to predict what the effect of manipulating that target will be. Now, there's many limitations of Mendelian randomization. People are interested in the topic. There's two relatively recent reviews which cover these. A particular limitation used to be statistical power, but these very large-scale genome-wide association studies are helping overcome that. As they are, the problem of not actually having a genetic variant related to your exposure of interest as more and more genome-wide association study results on traits are being produced. The major issue is pleiotropy, is that your genetic variant each individual genetic variant might be having an effect on other outcomes or the outcomes through mechanisms which aren't the one you're looking at. So one can address this by picking variants, for example, for a protein which in the gene related to that protein, which plausibly their main influence would be through that pathway, or taking many genetic variants from across the genome that influence an intermediate phenotype or exposure you're interested in, and comparing the effects across them, because it becomes decreasingly likely that you would get exactly the same answer through using different variants, because they won't all have the pleiotropy which perfectly balances across them. That would be unlikely. And there's methods which are discussed in these reviews which actually allow testing of the assumptions. But one method for assumption testing is by interacting your genetic variant with another endogenous factor, which means that the genetic variant has a larger effect on your exposure in one group than the other, with the prediction being that if the genetic variant effect is through your exposure, you should see a greater consequent effect on the downstream effects in the group who are having most response to the genetic variant. So here's one example of that, looking at the vexed question of alcohol intake and health. I mean, there's been huge amounts of publicity about the supposedly beneficial effects of alcohol for cardiovascular disease risk. You know, the famous U-shaped curve, that sort of moderate drinking was good for cardiovascular disease risk. Spoiler alert, it's not. That's really bad news. But you can address this by Mendelian randomization. It's a very difficult area to study because, of course, you get reverse cause. Whatever you're ill with, your doctor tells you to drink less. You get informative misreporting. You know, this guy says he's having 1 glass of alcohol a day, and that's what he puts on your questionnaire. And of course, it's heavily confounded. And it's not confounding the way you necessarily think. I mean, middle class people actually drink more than poor folk. Poor folk more likely to have concentrated periods of drinking, but the middle class drink through the week. So you know, the confounding isn't all that it predicts alcohol will have overestimated adverse consequences, as I will show. So these are the people who bought you vitamin E protects against coronary heart disease. I have got high blood pressure and I like a drink or two, so I thought this sounds like really good news, but is it true? So you can study this through Mendelian randomization by having genetic variants related to alcohol intake. Now, in cartoon form, if you drink alcohol, this is metabolized by alcohol dehydrogenase to acetaldehyde. And acetaldehyde gives you all the adverse consequences of drinking, so sort of flushing, palpitations, headache, guilt. Actually not guilt, but it gives you all the other adverse effects. And then that's cleared by acetaldehyde dehydrogenase to acetic acid and excreted. Now, in East Asian populations, Japan, Korea, Vietnam, China, etc., 30% of folk or so carry at least one null variant for acetaldehyde dehydrogenase. And if you have just one null variant, you build up much higher levels of acetaldehyde given a single or 1 or 2 drinks. If you have 2 null variants, your acetaldehyde levels are sky high, and people with 2 null variants really very rarely drink anything at all because it's unpleasant. And there's data on huge numbers of people, but this is from nearly 10 years ago. It was a meta-analysis of studies that looked at ALDH2 and alcohol intake and blood pressure to address the question of alcohol intake and blood pressure. And in men in these populations in East Asia, the men who had 2 null variants drank very little, the heterozygotes tended to drink an intermediate amount, and the homozygous wild type drank the most. So you got sort of dose-response association between genotype and amount drunk in the men. In these societies, when these studies were done, the women were drinking virtually very little, whatever their genotype. So here you have a lovely test of whether the effects of the genotype are actually all mediated through alcohol. Because if there's a pleiotropic effect, which isn't through alcohol, then the women should show that effect just the same as the men. If the effect is actually through alcohol, and what you're seeing is an alcohol effect, i.e. the Mendelian randomization is working, then you should see effects in men but not in women. And I said I already gave you the spoiler alert. Here it is. The men who carry 2 versions of the null variant, who drink virtually nothing, have very considerably lower blood pressure than the men who have 2 copies of the wild type variant. So alcohol actually increases your blood pressure, and if you've got high blood pressure, then take your ACE inhibitors, then you can still have a drink or 2. But importantly, in the women, the genetic variants are not associated with blood pressure, which provides evidence that this is not a pleiotropic effect. This is the gene itself is not having the effect. This genetic variant, which is out of interest, this is the largest effect genetic variant there is on blood pressure, common variant that's ever been identified. And one of the nice thing about this is its effect is entirely environmentally mediated, through a modifiable exposure, i.e. alcohol. And studies have now been done which suggest that across the board, alcohol unfortunately does not have a beneficial effect on cardiovascular health. In fact, has a detrimental effect across measurable levels. Here's a second example of that gene-environment interaction. When genome-wide association studies were done of lung cancer, the top genetic variant in 3, which reported at the same time, was in a nicotine receptor, which related to heaviness of smoking. And if you started smoking, you found it harder to give up if you carried these variants. So that was the top hits. So again, the top genetic variant for lung cancer is one which its effect is entirely through the environment, through smoking. So Mendelian randomization shows that smoking does indeed cause lung cancer. But here we were looking at the effect of smoking on body mass index, because people, especially people who carry on smoking, say that it helps regulate their weight. It actually works to keep weight down. And sometimes there's been editorials in the BMJ saying, having to dismiss the myth of weight control through smoking. So these people are sort of everyone's just lying to us. Everyone who thinks they know what the effects. The truth is that smoking does indeed help suppress appetite and keeps your weight down. And this is demonstrated here by looking at the genetic variant related to more heavier smoking, unrelated to BMI of people who've never smoked. So the genetic variant itself isn't having the effect. But in people who've ever smoked, and amongst ever smokers, the ones who carry the variant inhale more, smoke more, that can't give up, they are of lower weight. So smoking has a causal effect, a non-trivial causal effect on body mass index, and this might be part of the reason why body mass index appears to protect against lung cancer. Mendelian randomization can be useful in situations where you can't really envisage doing randomized controlled trials. So here I'm considering the effect of maternal alcohol intake during pregnancy and offspring outcomes. Studies were done based on the Millennium cohort, which suggested that rather like the U-shaped curve with cardiovascular disease, you saw a U-shaped curve for maternal drinking. It was best if the mothers were sort of moderate drinkers. The baby boys were less badly behaved, were higher IQs, did better at school, etc. Light drinking, no risk to baby, say researchers. Maybe good for baby boys. I was in India at sabbatical when this came out, and I love this picture of the pregnant woman drinking a glass of wine. Says, "Mom's light drinking doesn't harm baby." And then one more news story: pregnant women told to ignore drinking advice. So which advice are you being told to ignore? The advice to drink more, or the advice to drink less? Rather confusing situation. And here are the data from one of those papers. And there you are. There's the U shape of behavioural difficulties in the offspring. Light drinkers look as though they're doing the best. Now, not actually plotted or straightforward in the paper. You have to extract this information from supplementary tables, etc. I plotted a couple of other factors like smoking by drinking. There you go. The light drinkers do best there, or socioeconomic deprivation. The light drinkers do best there. So the shape of all the confounders is exactly the same shape as the outcomes. And the implication here that you might as well have a mother who's a binge drinker as a never drinker, it might not be causal given that confounding, but adjustment doesn't do any good. Now, in European populations, unlike East Asian populations, they don't carry ALDH2 variation, but they do have variation in alcohol dehydrogenase, which influences drinking, but to a less dramatic extent. But these are the data from Louisa Zuccolo looking at the mothers in a study birth cohort called ALSPAC, and their drinking during pregnancy. This genetic variant was related to about a twofold difference in drinking more than 1 drink per day and a twofold difference in the other direction for never drinking. So it related to a sort of leftward shift in the drinking pattern. Now, if Louisa did the observational analyses in ALSPAC, and what she showed was the same as in the Millennium cohort, more frequent drinking in pregnancy seemed to actually be good for the kids in terms of their school performance. And you adjust for everything. And ALSPAC has amazing data. You can't get a better data set for adjusting for all the confounders you can think of. But if you do the Mendelian randomization, i.e. looking at the genetic variant the mothers carry during pregnancy that influences their drinking, the results are diametrically in the opposite direction. i.e. they suggest maternal drinking during pregnancy is associated with worse outcomes, and that this is the same result is seen if you exclude the women who report ever having an episode of binge drinking. So the observational evidence, in my view, this is the strongest evidence, and the observational evidence suggests your mothers should drink is unlikely or less likely to be true, to be real. I'll actually skip that. So I'll move on to talking about exposure measurement and prediction. I'll very briefly talk about some epigenetic factors. So other ways of trying to improve evidence about things which might influence population health. So of course genes have to be expressed to have effects. And part of that process of the genes being expressed is their regulation through a whole host of epigenetic processes, of which the one which is most tractable to study and has the stability characteristics that mean it has long-term effects is DNA methylation. This is sort of on top of your DNA string is methylation, which influences the expression or non-expression of that part of the DNA. And this can be measured, so methylation can be measured across the genome and has, as I say, reasonable stability. And it is also responsive to the environment. So it's thought to be a potential mechanism through how the environment generates long-term differences in gene expression and therefore long-term differences in biology. And it can be used as an exposure indicator, diagnostic or prognostic aid, or might be a mediator. Now, tobacco smoke has dramatic effects on the epigenome, and they are long-lasting. And combining these measures allows you to predict people's smoking behaviour, long-term smoking behaviour, as I'll go on to show some more data on that. Also, and fascinatingly, you can predict intrauterine exposures to maternal smoking through these markers, and I think this is really quite remarkable. These are some ROC curves looking at predictive ability of whether one's mother smoked. Now, of course, if your mother smokes for socioeconomic reasons and cultural reasons, offspring are a bit more likely to smoke. So if you look at the epigenetic or methylation profile related to own smoking, your own smoking. It does actually predict whether your mother smoked. But the specific markers related to maternal smoking, some of which aren't related to your own smoking because they're happening during the fetal period, there's different biology going on. Those markers which predict maternal smoking up to the age of 50 or 60, they almost certainly carry on predicting after that, it's just no one's measured it. You can just say whether someone's mother is a smoker. Now, if you think of the sort of conditions we're trying to look at and to think whether maternal smoking has effects when people can't really report whether their mothers smoked or not, or you might just have got biological samples and no questionnaires, you can actually use the methylation data to predict that. That's a rather remarkable phenomenon. We wrote an editorial about these sort of processes but which we refer to as a sort of biosocial archive. And it was called Stem Cells to the Law Courts because in the US there's a few cases which haven't gone through, but where children looked as though they were going to sue their mothers, for damaging them while they were pregnant. In fact, years ago, decades ago, literally, when I did a first study on maternal smoking, my mother, who had been a sort of 2-pack a day woman, and 2 glasses of Guinness a day while she was pregnant with me, so to sue me, that was her response to me. But you can also use people's own methylation data as an index of their own smoking, and there's a huge number, as I say, a huge array which you can put together. The top factor is this aryl hydrocarbon receptor repressor, AHRR, which is related to detoxification of toxic factors, amongst other things. But you can actually predict better if you measure the lot. But just with this AHRR, just with this regional methylation, we, with colleagues in Copenhagen, Stig Bjørsen and others, were involved in looking at how this predicted lung cancer. And firstly, it was a very good predictor of smoking. And the thing, in former smokers, you could separate out former smokers and the pack years the former smokers had had. It related to time since giving up. And people who gave up smoking 20, 30 years on, you could still see a marker that they had at one stage been smokers, and the heaviness of smokers in current smokers. And it's a very strong predictor of lung cancer, much better than just people's smoking histories. Indeed, if you put in the methylation measures of smoking, reported smoking almost dropped out of the sort of prediction, which is sort of remarkable. But there's arguments both ways about CT scanning for early intervention through early detection of lung cancer. But this would add very strongly to the predictive model, better than the current predictive model, and from a simple blood test, just looking at one sort of methylation site. So it can be very useful for exposure indication and for prediction. And we're very interested in trying to look at this methylation as a marker of indoor air pollution in women in India, for example, who sit over stoves for a long period of time, to see where you haven't got a good biomarker of that exposure. But and the estimates of how important that is in terms of COPD in never smoking women in those countries is debated, and the epidemiology requires good biomarkers. But you don't believe everything you read in the sort of DNA hype. This company wants to analyse your saliva to try to predict when you'll die. And indeed, there is there is a marker of epigenetic age. You can use methylation measures to predict people's age, and it predicts it reasonably well. That's why we talked about the forensic epigenome. If you leave a drop of blood at a crime scene now, they can say, this is about sort of mid-50s, smoking, heavy drinking male. I mean, all crimes committed by mid-50s. So, but anyway, you can actually say what sort of roughly the age is. But it works. It doesn't work quite as well predicting your death as asking someone their income, for example. So, so you don't need saliva for that. And DNA methylation could be a mediator and therefore targeting, and you can use Mendelian randomization to tell you whether the DNA methylation is actually part of a process going through the disease, and therefore could become in future a target for intervention. So this is a quite exciting area. But I was told I'm going to finish very quickly, so there's time for questions, but I'm going to finish with just a few sentences about, maybe sort of unexpected few sentences, but about how genetics tells us about the prevention paradox. I'm sure most of you here have been taught or read about Jeffrey Rose's strategy of preventive medicine, the prevention paradox being that lots of people have sort of small levels of individual benefit, if you could even attribute that benefit, but that overall has a big effect for the population. But also, and related to that was his famous distinction between sick individuals and sick populations, and that the causes of disease in individuals may be different to what causes the disease level in populations, which sort of sounds mystical, but it is actually supported by just old school variance decomposition genetics. We take twin studies and you decompose the variance into 3 components, one of which is just the additive genetic, is things just straightforwardly genetic. The second component is shared environment. That's the component that's shared by people who grew up together in the same household. Siblings, things shared by siblings. And then the third thing is non-shared environment. And for most diseases you look at, the interesting thing is very few diseases have no genetic contribution. Lung cancer, even though it's caused by smoking, has a genetic contribution. Some of which, at least, is through the genetic propensity to smoke, which there certainly is. Genetic propensity to smoke. Small shared environmental component, which comes from the fact that if people grow up in the same household, if the parents smoke, are more likely to smoke. There is a shared environmental component. But the largest component is this so-called non-shared environment, or unique individual, what are called unique individual risks. Now, this is sadly, I think this is a misnomer, because I think that calling this environmental, I think, is misleading, and it also leads to this sort of search to find these missing environmental factors that we haven't found. People quote these things and say, 62% of lung cancer. We've got to sort of think about these individual level factors. And the thing that we all used to put on our grants when we're trying to get money to do coronary heart disease research is the famous sort of Whitehall study analysis, saying that 2/3 of the risk difference between groups isn't explained. And as you got more desperate to get money, it's become 3/4 of the risk isn't explained. And we're going to find all these new risk factors. But my view is that that these are basically, this non-shared environment, a big chunk of this is just stochastic processes, somatic mutations occurring very early on in development. It's just sort of, in some ways, bad luck, or somatic stable epigenetic changes, or just unique environmental, unique events during life, which we can't really think about as targets for modification. So I was going to give a thought experiment, which is sort of adapted from Geoffrey Rose. Imagine, I come from Bristol, imagine that in Bristol everyone has to smoke 20 cigarettes a day from adolescence on. So when you wake up in the morning, you get given a pack of Capstan full strength, and you're not allowed to go to bed until you've been shown to have smoked all 20 of them. But in Bath, the police stop anyone smoking at all. Bath's 16 miles away. And then you follow up the places for 50 years. Now, of course, Bristol would have much higher lung cancer rates. But within Bristol, how does smoking relate to lung cancer risk? Everyone is smoking 20 cigarettes a day. Therefore, you obviously can't demonstrate that smoking has any influence on lung cancer at all. The factors that might show a difference would be genetic or occupational, or I think, but basically just luck, or a big chunk of it would just be luck. And within, so those are the factors which causes one individual rather than another to get lung cancer. So that's the individual level cause that Geoffrey Rose would talk about. But between Bristol and Bath, virtually every case is attributable to smoking. So at the population level, virtually every case is attributable to smoking. At the individual level, no cases are attributable to smoking. The two things really are, or really can be, orthogonal. So at a population level, an exposure may be responsible for nearly all cases, but account for little of the difference in risk between individuals. And that means why we get these sort of stories which people love. This is from the Sun. Winnie, she's lighting a cigarette from a 100th birthday cake, and it says she's been smoking for 93 years and she ain't quitting now. I used Winnie in a paper some years ago, but I came up with a better example more recently. Is Johannes Hiester, who was a German actor. This is a better example because he was an actor, and you can go back to photos of him in the 1920s smoking right the way through to when he was 106. And the thing about Hiester is that it's very difficult to find a picture of him without a cigarette, actually. Now here he is. He quit smoking at age 106. I did it for love, for my wonderful wife. She should have me for as long as possible. I think if you look, his wife does look a bit younger, but if you look closely, I think she's had some work done. But he lasted another 2 years. But you get these cases because the between individual factors are, these non-shared, so-called non-shared environmental factors are largely stochastic, and we have very good evidence that that there's a major contribution of stochastic processes to disease. One thing which is very obvious if you think about it is just cancers of bilateral organs. So consider kidney cancers or breast cancers, which have been studied most. If someone gets a breast tumour, breast cancer in one breast, then the probability of getting a second primary tumour in the opposite breast is elevated compared to people in the general population. But it's not that elevated. You know, it's a bit up, but it's not that dramatically up. Now, what sort of epidemiology could pick up what factor is leading to cancer in one breast, but those exposures are not generating the disease in the other breast? It's identical germline genome, identical exposure to circulating metabolites, identical exposure to the external environment, as far as the extent we can measure it. So, bilateral organs really just give us the limits of prediction, the limits of personalised predictive medicine, I think. And as I say, there's just these sort of subcellular events, which exactly when they happen. If it happens at the 4th cell division rather than the 8th cell division, you have orders of magnitude differences in numbers of cells which have had mutations. And the chance can be from the subcellular to the biographical level. So we can imagine Winnie, at the age of 62, the postman might have come at 11 a.m., knocked on the door. Winnie opens the door, blast of wind, and she coughs up some metaplastic cell. If the postman had come at 1 minute past 11 or 10:59, that wouldn't have happened. So those sort of events are not studyable. So it doesn't matter to me whether this is epistemological, i.e. we just simply can't know it, or ontological, it's truly random. Those sort of processes just aren't studyable, and that tells us that's the good news for epidemiologists, because it means populations make sense in terms of studying disease risk, because the individual prediction and ability to detect differences in risk is always going to be poor. And if any of you suffer from insomnia, this was the John Snow lecture in 2011. Paul mentioned that today is John Snow's birthday. I didn't know that, but this was the John Snow lecture in 2011 is on this issue of individual and population risk, which bothered me for 20 years. And as I say, perfect cure for insomnia. And thanks very much. And those are the people who did all the work.
Thanks, George. Thanks, that's a really brilliant overview. We've got time for 2 questions. Please give your name. This is being recorded on Panopta when you give the questions.
Gareth Lysett from the Vector Group. How far would the epigenetics go back? Does it go beyond the mother?
Yeah, that's a very interesting question. So there's an important distinction to be made between the epigenetic processes within a generation, and that within a generation is from when you were just a conceptus. During that period, the methylation is sort of stripped, is completely stripped, and it goes through a stripping and replacement process. So it's always been thought that most if not everything is sort of wiped clean across generations. And there's reasons to think that obviously you have to go back to being a sort of primordial type cell. You can't have all the differentiation which happens when cells differentiate. So the within generation epigenetics is entirely non-controversial. It's clear that processes have to happen. That you start with a cell which has pluripotency, can become anything, and the cells that become kidney divide. Once they become kidney, you get replacement of the kidney, and also many diseases which exist in organs which still undergo mitosis, cell division. The disease has to cross mitosis. So single generation epigenetics is involved clearly in development and is involved in disease and is very interesting. Sadly, from my perspective, a lot of the hype about epigenetics has been about this sort of promise of transgenerational effects, where basically because of the sort of frisson of excitement of invoking Lamarckianism and appearing to go against central dogma. Now, this is a very interesting process in plants. Clearly, these processes are important, but you've got to remember plants don't have the separation of germ cells from from the soma, which mammals we do. So and the actual demonstration of these things in in mammals or in humans, there is weak, it's relatively weak. There's weak evidence really about, even if in principle, how often they can actually be seen in principle. And it's also very clear that they're not a major component compared to germline genetics and environmental impacts. But it's a very every time a study comes out, say your grandfather's diet influences your risk of dying of cancer, gets on the front page of the news, front page of the papers. So, but I think that's somehow some of the hype on the transgenerational effects have sort of discredited the term epigenetics has become almost synonymous with that. The Guardian ran a disgraceful article about why everything Darwin said was wrong or something. Its title was, on the basis of some very one small study, showing something, because it sounds exciting.
I think there are people waiting to come in outside. Is that right, Joe? Yeah, so we're going to have to close now. So thanks so much. We can have some discussions outside, but thanks very much to George coming to us today.